Hello, welcome to Biology, the scientific study of life. Mechanisms of post-transcriptional regulation. Transcription alone does not constitute gene expression. The expression of a protein coding gene is ultimately measured by the amount of functional protein a cell makes. And much happens between the synthesis of the RNA transcript and the activity of the protein in the cell. An increasing number of examples are being found of regulatory mechanisms that operate at various stages after transcription. Notice this figure. These mechanisms allow a cell to fine-tune gene expression rapidly in response to environmental changes without altering its transcription patterns. Here we discuss how cells can regulate gene expression once a gene has been transcribed. Notice also RNA processing. RNA processing in the nucleus and the export of mature RNA to the cytoplasm provides several opportunities for regulating gene expression that are not available in prokaryotes. One example of regulation at the RNA processing level is alternative RNA splicing, in which different mRNA molecules are produced from the same primary transcript depending on which RNA segments are treated as exons and which as introns. Regulatory proteins specific to a cell type control intron-exon choices by binding to regulatory sequences within the primary transcript. Notice here alternative RNA splicing. Notice also exons, DNA, primary RNA transcript, and mRNA splicing. Notice also the primary transcripts of some genes can be spliced in more than one way, generating different mRNA molecules. Notice in these examples that one mRNA molecule has ended up the green exon and the other a parable exon here. Notice that with alternative splicing, an organism can produce more than one type of polypeptide from a single gene. mRNA degradation. The lifespan of mRNA molecules in the cytoplasm is an important factor in determining the pattern of protein synthesis in a cell. Prokaryotic mRNA molecules typically are degraded by the enzymes within a few minutes of their synthesis. This short lifespan of prokaryotic mRNAs is one reason prokaryotes can vary their patterns of protein synthesis so quickly in response to environmental changes. In contrast, mRNAs in multicellular eukaryotes typically survive for hours, days, or even weeks. For instance, the mRNA for the hemoglobin, polypeptides A globin and B globin in developing red blood cells are unusually stable, and these long-lived mRNAs are translated repeatedly in these cells. Research on yeast suggests that a common pathway of mRNA broken down begins with the enzymatic shortening of the poly-A tail. This helps trigger the action of enzymes that remove the 5-cap notice that this helps trigger cap. The two ends of the mRNA may be briefly held together by the protein involved. Removal of the cap, a critical step, is also regulated by a particular nucleotide sequences in the mRNA. Once the cap is removed, nuclease enzymes rapidly show up the mRNA. Nucleotide sequences that affect the length of time an mRNA remains intact are often found in the untranslated region, UTR, at the free end of the molecule. In the one experiment, researchers transferred such a sequence from the short-lived mRNA for a growth factor to the free end of a normally stable globin mRNA. The globin mRNA was quickly degraded. During the past few years, another mechanism that blocks expression of specific mRNA molecules has come to light. Researchers have found small single-stranded RNA molecules called microRNAs, short-term miRNAs, that can bind to complementary sequences in mRNA molecules. The RNAs are formed from longer RNA precursors that fold back on themselves, forming a long double-stranded hairpin structure held together by hydrogen bonds, an enzyme fittingly called dicer, or called dacer. Dacer then cuts the double-stranded RNA molecule into short fragments. One of the two strands is degraded and the other strand, miRNA, associates with a large protein complex and allows the complex to bind to any mRNA molecules that have a complementary sequence depending on various factors. The miRNA protein complex then either degrades the target mRNA or blocks its translation. Inhibition or gene expression by RNA molecules was first observed by biologists who noticed that injecting double-stranded RNA molecules into a cell somehow turned off a gene with the same sequence. They called this experiment phenomenon RNA interference, or RNAi. It was later shown to be due to small interfering RNAs, or cRNAs, RNAs similar size and function as mRNAs. In fact, subsequent research showed that the cellular machinery that generates siRNAs is the very same as the responsible for producing mRNAs naturally in the cell. The mechanisms by which these small RNAs function appear to be similar as well. Because the cellular RNAi pathway can lead to the destruction of RNAs with sequences complementary to those found in double-stranded RNA molecules, it is commonly believed to have originated as a natural defense against infection by RNA viruses. However, the fact that the RNAi pathway can affect expression of cellular genes also supports alternative models. In any case, it's clear that RNAi plays an important role in regulating gene expression in the cell. Initiation of translation. Translation presents another opportunity for regulating gene expression. Such regulation occurs most commonly at the initiation stage. Notice at this figure. The initiation of translation of selected mRNAs can be blocked by regulatory protein that bind to specific sequences or structures within the untranslated regions at the 5' 5 UTR of the mRNA, preventing the attachment of ribosomes. A different mechanisms for blocking translation is seen in a variety of mRNAs present in the egg cells of many organisms. These stored mRNAs lack poly-A tails of sufficient size to allow translation initiation. At the appropriate time during embryonic development, a cytoplasmic enzymes adds more residues allowing translation to begin. Alternatively, translation of all the mRNAs in a cell may be regulated simultaneously in a eukaryotic cell. Such global control usually involves the activation of or activation or inactivation of one or more the protein factors required to initiate translation. This mechanism also plays a role in starting translation of mRNA that are stored in egg cells or NIG cells just after fertilization. Translation is triggered by the sudden activation of translation initiation factors. The response is a burst of synthesis of the protein encoded by the stored mRNAs. Some plants and algae store mRNAs during periods of darkness. Light then triggers the reactivation of the translation apparatus. Protein processing and degradation. The final opportunities of controlling gene expression occur after translation. Often eukaryotic polypeptides must be processed to yield functional protein molecules. For instance, cleavage of the initial insulin polypeptide, proinsulin, forms the active hormone. In addition, many proteins undergo chemical modifications that make them functional. Regulatory proteins are commonly activated or inactivated by the reversible addition of phosphate groups, and proteins destined for the surface animal cells acquire sugars. Cell surface proteins and many others must also be transported to target destinations in the cell in order to function. Regulation might occur at any of the steps involved in modifying or transporting a protein. Finally, the length of time each protein functions in the cell is strictly regulated by means of selective degradation. Many proteins, such as the cyclins involved in regulating the cell cycle, must be relatively short-lived if the cell is to function appropriately. To mark a particular protein for destruction, the cell commonly attaches molecules of a small protein called ubiquitin to the protein. Giant protein complex called proteasomes then recognize the ubiquitin target. Molecules and degrade them. The importance of proteasomes is underscored by the finding that mutations making cell cycle proteins impervious to proteasome degradation can lead to cancer. Okay, thanks for listening. Notice here this stage degradation of a protein by notice also protein to be degraded. Okay, thanks.